In this video we will discuss about the CAR T cells with bispecific protein, and there is a slight changes. There is a slight change in this bispecific protein. That is, they make a human version of this anti-PD-L1 and human interleukin 12. So they wanted to check whether their CAR T with human version will be able to work or not. And in this figure, they did the in vitro studies to validate or to check their hypothesis whether the CAR T with bispecific human version proteins, that is human version anti-PD-L1 and humanized interleukin 12, can work against the humanized ovarian cancers or not. So in the panel A, they shows this humanized expression levels of this humanized interleukin 12 and this FC levels, humanized FC levels. So what they did is this time they introduced this FC region in the interleukin 12 and PD-L1 so that it will increase the half-life period within the humans. So, but they did their studies in the in vitro system. So the experimental plan is shown in the panel B. So where they took the humanized cancer cells that has this TAC72, and in order to make these tumor cells express PD-1, they introduced this IFN gamma in the conditioned media, or they use the IFN gamma conditioned media for overnight to grow these human PC3 cells so that the tumor cells will express PD-1, and thereafter they add the supernatant from the engineered T cells. So what these engineered T cells are? These T cells are the Jurkat cells that are transduced with their CAR T with bispecific humanized version of anti-PD-1 and human interleukin 12, and then they took the supernatant and added it to these tumor cells, and after 1 hour they check this tumor killing and the other immune cells. So first they check the expression levels of this PD-1, and we can see that expression levels of the PD-1 are seems to be high in the IFN gamma conditioned media and the untransduced cells, and they are also high where the mutant anti-PD-1 is used. This is not mutant. They are also high where they use only human interleukin 12 FC, but when they use the anti-PD-L1, they are able to see a low amount of PD-1 for this only anti-PD-1 PD-L1 FC, and also with anti-PD-1 anti-PD-L1 FC and human interleukin 12s. And this could be because the anti-PD-L1 might be competing with the PD-1 expressed on the tumors, and that is the reason they are being masked when during the flow cytometry analysis. When they check the tumors FC region, or when they check the FC expressed on the tumors, they are able to see that the tumors expression levels are high when they use the anti-PD-L1 FC, and because the anti-PD-L1 is bind to the tumors, so they are able to see the anti-PD-1 binding to the tumors also is high, both in the case where they use only anti-PD-L1 FC and also in the case where they use anti-PD-L1 FC and human interleukin 12. And thereafter they check the human interleukin 12 expressions on the tumors, and it is highest in the case where they use anti-PD-L1 and hIL-12. And it is surprising that human interleukin 12 levels are low in the case where they use only human interleukin 12 FCs. And this could be because the PD-1 might be creating problems for this human interleukin 12 to bind to the tumors, and which in the last case is being controlled by anti-PD-L1, and thereby the human interleukin 12 can bind to the tumors. And then they check before going into the killing assay, they wanted to check whether the transduction efficiencies in different conditions are similar or not. and they are able to see the transduction efficiencies are almost similar, except in one case where TAC72 only anti-PD-L1 FC is used, where it is around 38%. Otherwise, in all cases, the transduction efficiencies are more than 40 and less than 50. And then they also check the intracellular human interleukin 12 levels, and these levels are high in the case where human interleukin 12s are also used, so indicating that their system is working as per the expectations in terms of the translations. Then they check the FC expressions also, and they are able to see the FC expressions also of the same levels, that is in the case where human interleukin 12 FC is used or anti-PD-L1 FC along with hIL-12 is used. With less amount of this anti-PD-L1 FC is found in the intercellular part, because these anti-PD-L1 FCs are mainly secreted into the extracellular matrix. So thereafter they check the tumor cell killings, and the tumor cell killings, the maximum tumor cell killings are found in the case where they use both bispecific proteins and the CAR T cells targeting TAC72. And then they also check some immune cells, that is CD45 plus, CD19 plus cells. They are also high, or the T cells expansions are also high in the J case. So in the I case, they checked CD45 plus, CD19 plus CAR counts. So CAR counts are also seems to be high, and the CD25 plus cells are also high. And these highs are maximum in the case where they use both bispecific proteins along with CAR. And IFN gamma levels are low because the anti-PD-1 because this because the PD-1 that are expressed on the tumors are being masked by the anti-PD-L1 through this supernatant of the engineered T cells. That could be the reason why the IFN gamma levels are low in that case, but they are high in the case where they use only human interleukin 12 FC. Then they check the tumor cells PD-1 expressions. They are almost similar in all the cases, or we can say that there is no statistically different compared to the they are not statistically different in all the 4 cases, and they are high compared to the control. And the CAR T PD-L1 expressions, CAR Ts are not naturally express PD-L1, but these expressions of this PD-L1 could be the anti-PD-L1 that are transduced, for which these CAR T cells are being engineered. That could be shown here in the panel M. In the panel N, and they are saying the tumor cell surface or the percentage of FC. So they can see that the tumor cells that are found, or tumor cell surface with percentage FCs are high in the case where they use only anti-PD-L1 FCs, because these are bind to the tumors. But in the case combined case, they are able to see less amount of this percentage FC, because some of these cells are dead, or the dead cells in the last case is high in the compared to the case where they use only anti-PD-L1 FC. And when they check the tumor cells human interleukin 12, they are maximum in the case where they use both bispecific proteins. And similarly, CAR T cells human interleukin 12 expressions are also maximum in the case where they use both bispecific proteins. And these are low, interleukin 12 expressions are low in the case only human interleukin 12 is used, because in this case the cytokines forms by the cytokines, or we can say IFN gamma forms by the these tumor cells could be affecting these human interleukin 12 expressions. And also this PD-1, since it does not have the anti-PD-L1, the PD-L1 that acts on the tumors might be deactivating this uh, T cells or these CAR T cells. That could be one of the reasons, but they have not validated these points in this paper. This is just my own understanding. This is just based on my own understanding. So I hope you understood how they validate their CAR T with bispecific humanized version proteins. And finally, they conclude that since the results are optimistic, their engineered CAR T with bispecific proteins could be moved to the clinical translations. This is their final verdict. I hope you understood this paper and the results that are presented in this paper. If you need to understand more about this paper in detail, you can go through the reference that has been provided in the description. You can also go through or glance upon the gist that has been provided in the descriptions to understand the specificity of some of the experiments. So that's all for this video. Thank you for your time.